Hello students, today we discuss the next question in your topic plant tissue culture, that is describe the aspects of micropropagation with reference to orchids. So in last videos I talk on the about the micropropagations in floriculture. So this is one of the aspects of the micropropagation, that is uh, with reference to the orchids. So first of all you have to know about the orchids. So what are orchids? So orchids are the beautiful flowering plants which are used as a ornamental plants. So these orchid plants are the very beautifully flowering and that are used in the ornamental purposes. Its family is the Orchidaceae. Uh, it contains about 1000 genera and more than 22,000 species. So these orchids contains about 1000 genera and 22,000 species. These plants are terrestrial or epiphytes. So some members of orchids are the terrestrial. Terrestrial, that means they are growing on the lands, and some plants are epiphytes. Epiphytes, that means the plants or orchids which are growing on the other plants. For example, on the big trees like mango, jamun, on these trees, some orchid plants are growing. These orchid plants are remains as 15 to 20 days, and certain orchids flower blooms and remains fresh for the 13 days. So जो prolonged keeping quality of these orchid flowers are uh, very strong. So these orchid flowers can be remains after 15 to 20 days, or some flowers of these orchids will be up to the 30 days. So um, these prolonged period of this period, or the flowers will be keep as it is up to the 15 to the 13 days. That's why these plants or these flowers of orchids are used for the ornamental purposes. So what are the importance of orchids? So its main important, it is used for the commercial purposes, because due to the they will be keep as it is up to 15 to 13 days. It is used for the ornamental purposes. These flowers of these plants will be very attractive and remains fresh for the longer durations, and shows mimicry with the animals, insects. The aesthetic value of the orchid flowers made them highly valuable in the commercial. This flower shows the mimicry with animals or insects. That means these flowers, some flowers of orchids are look like the animal faces. That means some look like the these uh, flowers are shows like the faces of the monkeys or some insect, etc. So they showed some mimicry of these animals as well as the insects. Also this flower got some aesthetic value. That's why they are used for the commercial purposes. Then second importance of these orchid plants is the perfume industry. That means these orchids are highly fragrant. These are too scented. Therefore, the orchids are useful in the perfumery industries. And due to the uh, uh, sense essence or the beautiful essence, and these flowers are used in the perfumery industries. Then next importance, it is the medicinal values. Some plants or some orchids are having the medicinal value because these orchids have been used in the source of herbal medicines in China since very 28,000 BC. It has medicinal value to cure many diseases and ailments. So these orchid plants also used for preparing the herbal medicines, and these orchid plants are very useful in many diseases. So these ultimately these plants are got the medicinal values. Then next importance is food value. The fleshy parts of the flowers, then enormous pollination material, that is pollen grains, gynoecium part are used as a food for insect, butterflies and humans. So due to the fleshy nature of flowers, these use or the some floral parts like pollen grains, gynoecium are used in the food for the insects, butterflies and also the humans. So ultimately they got the food values. So these are the importance of orchids. Next is about the technology for orchid production. So naturally the orchid production, there are two ways. One is the conventional farming and one is the micropropagation. Conventional farming, that means the traditional ways for cultivation the orchid plants. So by using the seed, root, stems, these plants will be cultivated. So in that some natural methods are also some artificial, that means some epiphytic orchids, and they are need of the epiphytic, that means they have used some bases. So in artificial techniques, the bark of the trees are used, and on these barks the epiphytic plant or epiphytic orchids will be grow, and some are naturally growing on the plants. So this is the conventional method. In micropropagation, by using the tissues of orchids, the culture to grow, and we get hundreds of or thousands of plants by using the tissue culture technique. As you already discussed in the micropropagations of the flowering plants, in that we have used the, first of all we use the or select the plants, select healthy plants. Then on these plants we can select the explant. The explant, that means the part of the plant which are growing on the nutrient medium. Then next technique is the sterilization. So sterilization of the plant material as well as the instrument is very important task. Then in the sterile condition we can inoculate these explant on nutrient medium. Then inoculated test tubes or slants will be transferred on the incubation chamber. Incubation chamber is nothing but the light and humidity or sterilization control room. In that control room there is the growth of explant on the callus. Then again we can reculture this callus on the different separate test tube in sterilized condition. So from one callus we can got 100 to 1000 number of plantlets. Then after development of shoot and root, these plantlets will be transferred on the pots, and then the hardening and cultivation of these micropropagated plants. So a similar technique is used in the orchid plants. So by this we can easily got the 1000 number of plantlets by using the micropropagations. Then the different micropropagations with reference to the orchids, that means and there are different types of orchids, and they are used for the micropropagation. For first one is the importance of orchids. So in that, bulbophyllum genus, then epidendrum genus, dendrobium, then pleurothallis, vanilla, and then anthurium, and some ornamental orchids. They can be used in micropropagation techniques and got the number of plantlets, and then they will be commercially marketed these plants. So here gives some examples that can be propagated through the, or they can be cultivated through these micropropagation techniques. For example, some bulbophyllum genus, some epidendrums and dendrobium है, pleurothallis है, vanilla plants है, anthurium orchids. Then the advantages of orchids. So you have to write same from question 1 of the floriculture. In that I already discussed about the advantages of floriculture. Same advantages you have to write in the orchids. So in that we have discussed about the micropropagation. So from a small amount of plant material we can get the 1000 number of plantlets. Then next advantage or importance of the orchids, you have to write the, it is disease free plant production, and also it got the superior plant production through this technique. There is the next advantage is the no variations will be observed in the micropropagated plant. That means the same plant is observed like the mother plants. Then next advantage, it is overcome to dormancy, because the generally the orchid seeds are normally they are dormant, they are not easily germinate. So due to this micropropagation, these seeds will be easily grow, and they can be more beneficial for the orchid plants. Then these also helpful for the early flowering. Due to this micropropagation, the normal period of the plant will be decreased, and the flowering will be earlier than 18 to 20 months. Then it is next advantage, it is control condition. That's why the orchids like orchid plants will be grown on the control conditions like nutrient, temperature, water, etc. This micropropagation will be helpful to improve the quality and quantity of produce. That's why commercially it will be helpful. Then next you have to write about the techniques of micropropagation which is used in the orchid production. These are also same from flowering questions or flower plant questions. First technique, it is the selection of plant material, and then next is the explant preparation, then sterilization, inoculation, incubation, then reculture of callus, then reculturing of callus, then hardening, and last one the cultivation. So these points I already discussed with you. So these all points will you have to write in the questions of the uh, describe aspects of the micropropagation with reference to the orchids. So this is all about the orchid micropropagation. I hope all of you are understood about this point. So now I stop here.